Chemogenetics is a powerful technique that allows researchers to control the activity of specific neurons using designer receptors and synthetic drugs. Here's how it works, step by step. It starts with designer receptors exclusively activated by designer drugs, or DREADDs. These are engineered versions of G protein coupled receptors, GPCRs. Proteins that sit on the cell membrane and normally respond to natural neurotransmitters like acetylcholine. In chemogenetics, these receptors are modified to no longer bind natural ligands. Instead, they are designed to be activated only by synthetic compounds that don't naturally occur in the body. The first step involves delivering the gene for a specific DREADD into targeted neurons. This is typically done using viral vectors, such as adeno-associated viruses, which are engineered to carry the DREADD gene without causing harmful effects. Once inside the neuron, the DREADD gene is incorporated into the DNA, and the cell begins to produce the receptor, embedding it in its membrane. The most commonly used DREADD is the HM3DQ receptor, a modified version of a muscarinic acetylcholine receptor. When activated by a synthetic drug like clozapine N-oxide or CNO, this receptor stimulates the GQ signaling pathway. Here's how it works in detail. CNO binds to HM3DQ, causing a conformational change in the receptor. This activates the GQ protein, which splits into two subunits, G alpha and G beta. G alpha then activates phospholipase C, an enzyme that cleaves a membrane phospholipid into inositol triphosphate, or IP3, and diacylglycerol, DAG. IP3 then binds to receptors on the endoplasmic reticulum, causing the release of calcium ions into the cytoplasm. This calcium influx excites the neurons, increasing its activity and making it more likely to fire action potentials. In contrast, another DREADD, HM4DI, is designed to inhibit neurons. When activated by CNO, it triggers the GI signaling pathway, which inhibits adenylyl cyclase, reducing levels of cyclic AMP. This decreases neuronal excitability, effectively silencing the neuron. To apply chemogenetics, researchers deliver CNO or other synthetic ligands through injection or systematic delivery. These compounds cross the blood-brain barrier, bind to the DREADDs in targeted neurons, and modulate their activity. This allows precise control over neural circuits, enabling researchers to study behaviors, diseases, and brain functions in real time. So, in summary, chemogenetics combines genetic engineering and synthetic pharmacology to activate or silence specific neurons via engineered receptors and designer drugs. Its precision and flexibility make it a revolutionary tool for understanding the brain and developing therapies for neurological disorders.